Hi everyone, today we're going to talk about activation of target cell receptors by insulin and the resulting cellular effects that occur after that. So first of all, what is the insulin receptor? So it is alpha to beta to heterotetramer. What does this mean? It means it's a tetramer that is made up of two alpha subunits and two beta subunits. The alpha subunits, they are on the outer surface of the cell membrane, whereas the beta subunits, they are integrated in the cell membrane. Now all of this system is connected with the help of these disulfide linkages. Now, how does the entire mechanism take place? So first of all, insulin, which is the hormone, it comes and it binds to the alpha receptor, the alpha subunit of the insulin receptor. And because of the disulfide linkages, the beta subunit ultimately also gets autophosphorylated. So when autophosphorylation of beta subunit takes place, there is another enzyme which is attached in the inner surface to the beta subunit. That enzyme is tyrosine kinase. This tyrosine kinase, it gets activated. What's the function of kinase? Kinase basically phosphorylates any other substance or any other molecule that's present. So tyrosine kinase is going to phosphorylate IRS, which is insulin receptor substrate. These are basically intracellular enzymes which are present within the cell, of course. And tyrosine kinase, it's going to phosphorylate IRS, the intracellular receptor substrate. This entire group of enzymes is going to get phosphorylated and activated, and it's going to help with the metabolism of glucose, with the metabolism of fat and protein. And also, there are vesicles which have glutes within them. These glutes will be, this vesicle will be forced to translocate, and it's going to fuse with the membrane here, and the glutes will be incorporated into the cell membrane, and that's how the glucose will come in. So this is the entire receptor of insulin. So this is the receptor, and what happens is that this entire system is known as enzyme linked receptor. What is it? It's enzyme linked receptor. Let me just jot it down quickly. Enzyme linked receptor. And how would I explain this? So basically, what's the enzyme here? What do you think? It is tyrosine kinase. So this is the enzyme, and that's the receptor. So basically, the receptor which is linked to the enzyme. So enzyme linked receptor. Again, this is alpha to beta to heterotetramer. There are two alpha subunits, two beta subunits. So first of all, insulin binds to alpha subunit of its receptor, which causes autophosphorylation of the beta subunit receptor, which in turn induces the tyrosine kinase activity. The receptor tyrosine kinase activity begins a cascade of cell phosphorylation that increases or decreases the activity of enzymes, including insulin receptor substrate. So the intracellular enzymes, their activity can either be enhanced or it could be decreased. And also another thing, these intracellular receptor substrate, they mediate the effects of glucose, fat, and protein metabolism. Glucose, fat, and protein metabolism. And the other thing that they do is they also cause the translocation of glutes. So this is all about the receptor, right? Now moving forward, what are the cellular effects that occur as a result of the activation of the receptor of insulin? So the first thing first, you have a lot of muscle cells and adipose cells. All of them have these receptors. So in these muscle cells and adipose cells, the insulin receptor, it gets activated, and then the entire translocation of these glutes happen. This allows a lot of glucose uptake. So the first change that occurs intracellularly is this increased uptake of glucose, especially in the muscle cells and the adipose cells. The second difference that occurs is that the cell membrane, it becomes more permeable to amino acids. So a lot more amino acids can come in. And then there can be also increased permeability to potassium and phosphate ions. The third thing that happens is intracellular metabolic enzymes, they are activated. So this is a much slower effect and occurs over the period of next 10 to 15 minutes. So what happens is basically there is changes in the activity levels of many more intracellular metabolic enzymes. And one of the prime examples that we already discussed was IRS, which stands for insulin receptor substrate. And the other effect is again much slower effect, and that is basically change rates of translation of messenger RNA. So there's change rates of translation of mRNA. This is a more prolonged effect. So these are the four cellular effects: increased uptake of glucose by muscle cells and adipose cells, more permeability to amino acid, potassium, and phosphate ions, intracellular metabolic enzymes activity is increased or decreased, whatsoever is required according to the situation, and then there's change rates of translation of mRNA.